Hello everyone. Today we will be discussing about the week 4 contents of your NPTEL Immunology course for the June to November 2022 session. Uh, so so, so we had like in the lectures, if you have followed, you will see that sir has talked about isotype switching. So, what is isotype switching? In like any kind of immunoglobulin molecule, what we had studied in the last week, we had seen that there is VDJ recombination that gives which gives variety to the variable region of your immunoglobulin molecule, because of which it can identify several types of antigen. But apart from the rearrangement in the variable region, immunoglobulin molecules can also undergo isotype switching. So isotype switching is what happens in the constant region of your immunoglobulin molecule. So as we were saying that initially on the surface of B cell, IgM kind of antibodies are mostly expressed, but later on in your later stages of infection, IgM immunoglobulin molecules can get changed to different types of and different types of antibody molecules like IgD or IgG or etc. So during primary immune response, the first type of immunoglobulin molecule that is produced is IgM type, because during the gene rearrangement, the first chain that is present just after the VDJ segment is the mu chain, and mu chain is corresponding to the IgM type of antibodies. So the Ig gene is composed of mu chain as the first constant region domain or segment, and so IgM type of antibodies are first synthesized during any kind of infection, followed by IgD type of antibodies, because mu chain is followed by your D chain. So any kind of in the constant region domain, so first you have this VDJ part, followed by your mu part, followed by your D delta part, and then rest of the other antibody segments. So first you will get the mu chain, and so once the VDJ is recombined, you can recombine with the mu chain, and then you can get the IgM type of antibody. So also, different heavy chain constant region is known as an isotype. So based on differences in the constant region and the variable region of light chain and heavy chain, antibodies could be classified into several types of determinants like isotypic determinant, allotypic determinant, and idiotypic determinant. So what is isotypic determinant? Any kind of changes in the constant region of both heavy chain and light chain. So say the mouse IgG antibody and IgM antibody, so that comes under isotypic determinant, and therefore the nomenclature as isotype switching that we use during this change of type of antibody from IgM to IgG type. And so this is known as isotypic determinants. So differences in the constant region of heavy chain and light chain will give rise to isotypic determinants, whereas differences in the constant region only. So here you will see that there are differences in the constant region of your heavy chain and light chain, but not the entire region. There will be certain parts of your or certain amino acids that will be varying in this constant region, that will give rise to different strains, different kind of IgG molecules only, not that your entire isotype is changed. Your mu chain is not going to become the G chain, but here your gamma chain only. The gamma chain will have certain differences in its sequence. So because of that, your different kind of IgG molecules will be different. So in case of like in different individuals of the same species will have different kinds of IgG molecules. So that is what is meant by allotypic determinants. And the last is the idiotypic determinants, which is basically the changes that are followed in the variable region of both the heavy chain and light chain. So it changes in this variable region or this CDR region or the hypervariable region. So those are associated with your idiotypic determinants. So idiotypes are basically different kinds of regions that is going to bind with, actually going to bind with your antigen, and therefore it it is very highly different from that of isotypic and allotypic determinants. And idiotypic determinants will give rise to variety of antibodies that can interact with a large library or large pool of antigens. So during secondary immune response, this isotype switching takes place at its maximum, and different types of antibodies are produced. Not only just secondary immune response, even during primary immune response as well. First antibody that is synthesized is IgM type, and then it gets converted to different kinds of antibodies. But mostly during secondary immune response, IgG is in the maximum amount. Okay, because IgG has a better affinity to your antigen. So better affinity to your antigen and better neutralization capacity. So IgG will be produced in larger quantities in your secondary immune response, and that is why your secondary immune response clears out the infection quite quickly. Now, how and why this kind of segments or rearrangement takes place? So, as I was explaining, so this is the VDJ part of your constant VDJ part of your heavy chain, and so just next to your VDJ part, so this is the variable region, and this denotes the constant region. So in the constant region, the first part is the mu chain, and the second part is the delta chain, or the which is going to correspond to the IgD type of antibody. So mu chain is again having different segments: mu1, mu2, mu3, etc. And delta chain also has different segments: delta 1, delta 2, delta 3, etc. So based on how the splicing, mRNA splicing, is going to take place, you can have either a secreted type of antibody. So this is a membrane bound type of your IgM molecule, and this is secreted form of your IgM molecule. So what determines the secreted or membrane bound form is this particular segment, that is S segment stands for secreted, and this M segment stands for membrane bound. So if your splicing takes place in a way that the membrane bound segments comes together, so this mRNA is going to produce membrane bound IgM molecules. But if the splicing takes place in a way that M molecules are excluded, just the S part is present, so here you will get the secreted kind of molecule. So this is again this is showing this is not secreted. So this is just delta IgM, delta IgD, sorry, the IgD type of antibody. So I will discuss about the membrane bound and the secreted form. So yeah, so S stands for secreted form and M stands for membrane bound form. And here the splicing is shown on the basis of the different types of antibodies that are isotypes of antibodies that are produced by splicing. So after VDJ recombination, if the mu part comes together along with the membrane M1 part or M2 part, this is the membrane bound IgM. And if the splicing happens between the delta 1, delta 2, delta 3 regions along with the membrane part, this is the membrane bound IgD. Now, whether it is going to be secreted or membrane bound, it is determined by this S region and this M region, as I was talking about. So, in all kinds of all types of isotypes of your antibodies, this secreted form and this membrane bound forms are present. So during mRNA splicing, if the secreted form comes in the final processed mRNA, so you will get the membrane, the secreted form of your antibody with a hydrophilic segment. So S stands for the hydrophilic, like S will be a hydrophilic segment, and it can be freely circulating in your blood. But if the membrane bound segment comes closer and is spliced together, then this M segment will come closer and will form the membrane bound part. So this is the membrane bound antibody, and it will have mostly hydrophobic residues to allow it to embed itself inside the membrane of B cell. So how, so this kind of mRNA splicing or mRNA splicing will result in membrane bound and secreted Ig molecules, immunoglobulin molecules. So what kind of amino acid residues are this composed of? So as expected, this stands for the secreted form of immunoglobulin, and this is the membrane bound form of your immunoglobulin. So you will see that mostly hydrophilic residues, this pink part is the hydrophilic residues. So hydrophilic residues are made up, makes up the secreted forms of your immunoglobulin molecules. Whereas in membrane bound form, there is a large portion of hydrophobic hydrophobic amino acid residues that is going to actually allow the immunoglobulin molecule to embed itself into the membrane. So in the membrane, hydrophobic residues are going to allow itself to embed. Also, apart from that, after transcription, polyadenylation is also very important. So polyadenylation sites are again present in this S region, just after the S region. So after polyadenylation only, your mRNA is totally ready to be translated. So mRNA splicing, mRNA processing is again another topic where you need to add the 5' cap as well as the 3' tail, and along with the exon intron splicing mechanism. So after all of that, after all of that, what is produced is the secreted form of antibody, or the membrane bound form of the antibody, and that is going to have this kind of residues. So apart from that, what kind of mechanisms we require for T cell activation is like T cell activation, naive T cell needs to interact with your antigen that is presented by your MHC molecule. And apart from that, it also requires a co-stimulatory signal where CD4 can interact with your uh, CD4 can interact with your CD4 or CD8, sorry, can interact. Here it is explained for CD8 plus T cell. Similar is true for CD4 T cells. So in case of CD8 T cells, CD8 is the co-receptor that needs to interact with your antigen presenting cell in order to have a proper immune response. So so antigen presenting cell which is presenting an antigen with the help of an MHC molecule will interact with your T cell receptor, and also it will be supported by the co-stimulatory signal like CD8. For here it is CD4, and after that, after it is finally recognizing this antigen, there will be more synthesis of cytokines and chemokines that is going to allow this proliferation of this single type of T cell. So single type of T cell will grow and proliferate, and this is known as clonal expansion, where this T cell with the particular T cell receptor is going to go and go and bind to your antigen of interest. So first step is antigen recognition. Antigen recognition can take place through the antigen presented by MHC molecule along with your co-receptor binding, which could be CD4 in case of T helper cells and CD8 in case of cytotoxic cell. Once it has recognized this antigen, it will get activated, and activation involves that this T cell receptor is perfect fit for this antigen. So this T cells will then synthesize or secrete interleukins, and these interleukins will help in the clonal expansion of the particular T cell molecule. This T cell will then go and become the effector T cells, and also memory T cells. So this is the same T cell that is going to be divided into two parts: memory T cell and effector T cell. So memory T cell will be lasting in your immune system for a longer period of time, and effector T cells is going to just go and attack the antigen and will kill the antigen producing cells or the pathogen. So this CD4 cells are mainly associated with the activation of macrophages, B cells, and other cells, whereas cytotoxic T cells are associated with cytotoxicity, and killing this target cells, it will release it can kill this target cells by cytotoxicity, by creating certain pores on the membrane and lysis of the cells. So CD4 cells and CD8 cells are like having different kind of functions, little different kind of functions. Uh, yeah, but the ultimate goal is the same, that to kill the pathogens. And then we can quickly go through that T cell receptor locus. So T cell receptor can be gamma delta type as well as alpha beta type. So gamma delta type of receptor is not very much in study, because like it is in study, it is not much known about gamma delta T cell receptor or what kind of antigen it recognizes, because most of the T cell receptors that are present inside our body is alpha beta type, and this alpha beta type of receptor is recognizing most of these antigens that are presented by MHC molecule. But see, gamma delta receptor, although it has a very similar structure, you can see that the structure is very similar to alpha beta type, but it does not require any kind of antigen processing. This kind of structure does not require any antigen presentation or antigen processing, and people are still studying that what kind of antigen could be recognized by gamma delta T cell receptors. Although it is hypothesized that it can bind to certain lipids, or like lipids of the pathogen, or of the invading foreign particles. And alpha beta type of T cell receptor is the most common type of T cell receptor. Now, all the T cell receptors also have this variable domains and the constant domain, similar to that of the immunoglobulin molecules or B cell receptors. And variable domains and V domains and C domains interact with the opposite domains of your MHC molecule. So V domain will yeah, V domain also has alpha like T cell receptors will have two chain. Alpha beta T cell receptor has two chain, mainly the alpha chain and beta chain. And gamma delta receptor has two chains, again gamma chain and delta chain. So with respect to the alpha beta receptors, the variable region of alpha chain will interact with the variable region of beta chain from the MHC molecule. So in the lecture also, if you have gone through, sir was talking about how you can how alpha chain's variable region interacts with the beta chain's variable region from the MHC molecule. This is the MHC molecule, and this is the T cell receptor. So T cell receptor is, as I'm repeating again and again, T cell receptor recognizes antigen presented only by an MHC molecule. So it is also recognizing this V variable region from the MHC molecule. Now, T cell receptors gene arrangement is also like, so it has two chains, basically an alpha chain and gamma chain with respect to the alpha gamma T cell receptor. So the alpha chain has again VJ regions. So VJ regions are present in this kind of so yeah. So this is in the light chain. Now they do not have that kind of concept like heavy chain or light chain like we have in case of B cell receptor, but alpha chain and beta chain is just like in T cell receptor we have an alpha chain and beta chain, but in case of B cell receptors we will have heavy chain, light chain, that kind of different, that kind of like comparison. So this is maybe you can compare this with the light chain alpha chain receptor. And so yeah, so in this alpha chain we have V region, V region as well as a J region along with the constant domain. And in the gamma chain we have this variable region as V and J. So there are certain repeats. So in alpha chain we have more. So sorry, this will not be light chain. This will be comparable to heavy chain, and this will be comparable to light chain. Why am I saying so is on the basis of the number of repeats that the variable V segment and J segment has. So you see V segment and J segment has a lot of repeats in alpha chain. So alpha chain has to be a lot of like a longer one than that of the gamma chain. So here we'll have V and J as 70 to 80 segments and 61 segments respectively. And apart from that, we have a constant region as well. In gamma chain, we have a V segment and J segment as 12 and 3 respectively. and constant regions. So what is important over here is the delta chain that could be present in this particular T cell receptor, the gamma delta receptor. So this delta chain is actually located entirely in the locus of an alpha chain. So this is the delta chain locus, and the delta chain locus also has instead of V and J, it has D and J segments, and this is located entirely inside the alpha chain. So to some extent, this is exciting because like gamma delta T cell receptors have certain portion of your gene present in your present in inside this alpha locus of T cell receptor. So this was this. And T cell receptor diversity accordingly, like what we have seen from the previous lectures, that T cell receptor diversity is mainly due to three reasons. First is the rearrangement in the variable region of heavy chain and light chain, mainly the VDJ segments and D and J segments in the heavy chain and light chain respectively. Also some kind of rearrangement in the constant region of heavy chain and light chain. So there can be T cell receptors diversity occurs due to rearrangement in the variable region, in the constant region, as well as due to addition of NNP nucleotides. I hope you know what is NNP nucleotides. So NNP nucleotides is what takes place when you are recombining with that RAG protein and TDT enzyme, etc., which is going to add palindromic and additional nucleotides to your between two segments of your gene. So if due to addition of this nucleotides, and this will be random addition, so we do not know what kind of nucleotides it's going to add. So random addition is going to increase the diversity of your T cell receptor. And there is another way where you can increase diversity, but this is only exclusively for B cells. So somatic hypermutation I had discussed in last class as well. So somatic hypermutation takes place on in rearranged T cell, where already the variable region and constant region is rearranged, but due to multiple infections by the same antigen, B cells that are having more affinity towards your same antigen is going to be selected each time during each infection, and that is going to basically increase your probability of the B cell to interact with your antigen in a better or in a strong manner. So somatic hypermutation happens only in B cell because antibodies need to interact with your antigen in a more stronger way. And this is the concept that is used in booster doses, that if you are injecting the same antigen again and again, your body will produce B cells with a better quality that can interact with your antigen more strongly. Now, there are certain differences that I picked up from the class, from this lecture that sir had discussed in NPTEL platform. So T cells are recognizing antigens which are presented by MHC molecule. What is MHC molecule? MHC stands for major histocompatibility complex, and it is expressed in the cells, and it is responsible for giving your cell a particular kind of like recognition that this is your body's cell, and this is like present like basically an address kind of thing, that this is your body cell and not a foreign cell. So MHC will identify MHC is basically used for identification of cells. And so there are certain differences which you can go through. So MHC1 and MHC2, the difference is MHC1 is recognized by T cells or cytotoxic cells, and MHC1 is actually expressed by all of your cells, mostly your brain cells, kidney cells, lung cells, etc. Every like most of your cells in your body is going to express MHC molecule. whereas MHC2 molecule is expressed mostly by the antigen presenting cells like B cells, T cells, and therefore MHC2 molecules is recognized by the T helper cells. Remember that T helper cells are responsible for activation of macrophages, B cells, and other cells, because T helper cells with its CD4 domain is going to interact with the MHC2 type of MHC2 molecule, and MHC2 is mostly present in your the immune cells, in the immune in your primary immune cells, in macrophages, dendritic cells, neutrophils, etc. Apart from that, MHC1, as the name implies, it has just one transmembrane domain. MHC2 has two transmembrane domain. Also, the structures were discussed in the lecture, where the peptide binding cleft, or why we are saying is that peptide binding cleft and not an antigen binding cleft, because MHC will bind only to a peptide and not to a whole antigen. So once the antigen is broken down into small pieces, the one of the pieces, which is a peptide, is going to bind to the to the uh, cleft, and it is going to have a different kind of segments. So MHC has again alpha chain, beta chain, etc. So in case of MHC1, it is made up of alpha 1 and alpha 2 chain, the peptide binding cleft, whereas MHC2, the peptide binding cleft, is constituted by alpha 1 and beta 1. So different kind of chains. Also, MHC1 can bind only to small length peptides, whereas MHC2 can bind to longer peptides. And MHC1 will bind to peptides which has certain residues, like tyrosine and leucine residues in its N-terminal and C-terminal ends, whereas peptides can interact through the entire length with its MHC2 molecule. So these are basically some differences between MHC1 and MHC2. And major histocompatibility complex in details, there will be lectures in the later classes. There maybe we can discuss about it in details. So that's it, and I have few questions. So if you are free, then you can just go through it and maybe answer it in the comment. Basically, I had already written the answers in the slide, so maybe, yeah. Uh, so do you like keeping it in polls or in slides, the questions?
Polls is better, ma'am. Polls is better.
Polls is better. Actually, I was thinking that because this lectures, these live classes are, I need to record it, and so I need to upload it in YouTube. And when you see it in YouTube, you will not be able to understand what question we are talking about if I'm giving it in polls. So that
Then,
is
ma'am, it is better as a question in call questions. Better this way as you gave it just now.
okay, 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 okay. So Rajkumar, you have a question?
Uh, Ma'am, one second, ma'am. YouTube link send it, ma'am.
I have actually sent it in the discussion forum. Did you guys get it?
Okay, ma'am. Not.
Yeah,
I don't know,
not.
ma'am.
Okay, in the NPTEL discussion forum where you can post all these questions and stuff like that. So there I have uploaded the slides link as well as the YouTube links for the last classes, three classes, three
Madam, we
classes.
have received the links by email or something. I had seen somewhere, but only up to week 3 we have received
Yes,
it.
yes, yes. Yeah, because week 4 is today. Like after the class only, I'll just upload it in the YouTube and then I'll just give it to you. Yeah.
Thank you so much, ma'am. Very well explained. Thank you so much, ma'am.
Thank you. Do you guys feel bored? Is it like okay, or do you want to make it more interactive?
You're going good, ma'am. You're going good.
Okay, thank you. So yeah, so this question's answer is okay. Somebody has asked that what is the variable chain is same for both IgD and IgM? No, the variable chain is also having this VDJ recombination, right? So it is going to be different in different kind of antibodies. So yeah, IgM and IgD. Okay, so if it is attacking the same antigen, then maybe it will have similar kind of variable region, IgD and IgM, but different antibodies will have different antigen will have different kind of antibody, different variable regions. Right. So T cells use all of the following for generating antigen recognition diversity on TCR. So combinatorial association of chains, different chains like alpha chain, the mu chain, delta chain, etc. This can be used for TCR. Also, the combinatorial association of segments. Segments are VDJ segments. The end region addition of nucleotides, as I was talking that, okay, some kind of nucleotides are added in the during cleavage by RAG1 and then addition by TDT. So those nucleotides are also going to add the TCR, add diversity to TCR. So, but somatic hypermutation is only restricted to B cells. So B cells will not, so T cell receptors will not undergo somatic hypermutation. So the second question is the locus encoding the TCR gamma chain is located. So I was discussing about this interesting thing, right? So yeah, gamma chain TCR is located in what locus? Okay, so yeah, correct answer is A. That's right. So this is a very easy question. First class of immunoglobulin molecule after activation of a B cell. Quickly, what is the answer? Just tell me, okay? Because I'm not able to see the chat
IgM.
box. Yeah, correct. So yeah, the answer is B, IgM. The second question, the fourth question, hinge region is present in all of the immunoglobulins except last class we were discussing about the structures, hinge region structures, and what kind of immunoglobulin will be present, like which immunoglobulin will not have hinge region. Can you just unmute and tell me instead of text writing in the chat box?
Option A, most of them have marked. I am sorry, I don't know the answer.
Okay, so option A is the correct answer actually. IgM and IgE does not have a hinge region. All rest of them has a hinge region. IgM and IgE, they are like little restricted in terms of binding. So yeah, that is option A is the correct answer. And finally, dimers of IgA, it is formed by what kind of bonds? Is it disulfide bond or alternate splicing or J chain or joining through a hinge region?
J chain.
Correct. So J chain is the correct answer. So IgA as well as IgM also has a J chain. So they are connected by a J chain. So Tanya, can you see my screen? Okay. So yeah, so Tanya had a question that of assignment 3, what would be the answer. So she had a confusion. So basically, what papain digestion does to an immunoglobulin molecule. So if I need to explain that, uh, so suppose this is your immunoglobulin molecule. Okay, this and this together forms the heavy chain, and this is the light chain. Okay, so let me use different colors. Uh, so your light chain has a variable region. Okay, and your heavy chain as well as has a variable region in them. Apart from that, it is also going to have a constant region, and similarly your heavy chain will have a this is a typical structure of an immunoglobulin molecule, where this is the variable region of your heavy chain. Okay, similarly this is the variable region of your heavy chain. This is the variable region of your light chain. This is also the variable region of your light chain, and these three are the constant regions of all of this, both the heavy chains. Okay, this 3 is the constant region of this heavy chain, this 3 is the constant region of the other heavy chain. So, and we know that immunoglobulin molecules, they are joined with each other with the help of disulfide linkages. So this is disulfide linkage between them. And what papain digestion does is it is going to cleave your disulfide bond. So you are going to get a cleaved bond. So you will have two FAB fragments, okay, and one FC fragment. Now what are you going to get in this FAB fragment? So if you are looking from this immunoglobulin structure, we are getting a dimer. So what is a dimer? Dimer is when two molecules are joined together, right? When two molecules are joined together. So this in this variable, in this dimer, in this FAB segment, so this is two dimers. So we have two dimers, and each dimer has a variable region from the heavy chain and variable region from the variable region from the heavy chain and the variable region from the light chain. Okay, each of this dimer has. Also, it has a constant region from the light chain, constant region from the light chain, and constant region from the heavy chain. So the question was whether papain digestion, what kind of dimers it is going to get. So yeah, so the dimer is like basically it is going to have a dimer of a heavy dimer of variable region of your heavy chain and variable region of your light chain, along with constant region of your heavy chain and constant region of your light chain. So this is like there was this option that was correct, option D, I think. So yeah, so this was like why dimer, why it needs to have the variable region and constant region of both heavy chain and light chain. So it contains both the variable region and constant region of both heavy chain and light chain. Is that clear, Tanya? So this so what is recombination signal sequence do? So it is basically a heptamer separated by a spacer sequence, separated and then comes a nonamer. Now, when you are having a kappa chain or a lambda chain in your variable region, this is in light chain of your immunoglobulin molecule. So you have several segments, say V1, okay, V2, V3, etc. So between these segments, you have this particular sequence. This heptamer, spacer, and nonamer sequence is present in between, say V1 and V2. So in between this region, it will be present. Now, what this sequence is meant for is, so there is a protein known as RAC1 and RAC2. So this is basically the recombination gene dependent enzyme endonuclease. Okay, gene dependent enzyme endonuclease. What it does is it will recognize, so it is, the name suggests that it is gene dependent. So it will recognize this particular recombination signal sequence, which is a heptamer space and a spacer. Spacer can be either 23 base pair or it can be 12 base pair. So what RAG1 and RAG2 will do is, RAG1 basically will recognize this particular sequence, heptamer, spacer, and nonomer sequence, and it will cleave it in between that sequence. And once it is cleaved, so this part will be cleaved. So what happens is it will help in recombination. So whenever V1 and between V2 there is a cleavage over here, and then we will have your there is another enzyme called TDT, that is uh, terminal, I think deoxytransferase, I guess. What is the full form? I forgot. Okay, so this enzyme will, once the cleavage is done, this enzyme is going to add a poly A tail to your ends, to the free ends of your V1 and V2. Now, RAG1 can recognize these sequences. So, heptamer, spacer, and nonomer is present in all of this, between the segment places. And so based on like where RAG1 recognizes, whether it recognizes between V1 and V2, or whether it recognizes between V1 and V3, so cleavage is done there. So if it recognizes between V1 and V3, this particular V2 will be cleaved out, and we will get only recombined V1, V3 kind of recombination. If it is recognizes this particular sequence between V1 and V2, so particularly this V1 I think will be cleaved out, and then we will get V2 and V3 as your recombined sequences. So recombination signal sequence plays a major role in allowing the RAG1 to identify a particular region and carry out recombination. So it is like it is a gene dependent enzyme endonuclease, and it is binding, it has a nonomer binding domain as well. This RAG1 protein has a nonomer binding domain, and that is why it is recognizing this particular sequence. So recognition signal sequences are meant for RAG1 to identify them and carry out the recombination in your light chain or as well as in your heavy chain. So that is what I had discussed. So see, location of recombination signal sequences, they are present like uh, between your variable regions, and see v lambda, v kappa. So there are this kind of v and j segments. Between those segments, this particular region is located. Look, so there are several kind of segments like this, not only just one. So there can be v kappa 1, J kappa 1, then B kappa 2, J kappa 2. So like those places. So it helps in recombining these segments together, and it causes variability in your immunoglobulin molecule. Did you get it, Rajkumar?
I get it, ma'am. Thank you, ma'am.
Okay. Okay, somebody has asked also explain about RAG. So yeah, RAG is a protein that is recognizing this particular recognition signal sequences, and it helps in recombination. Basically, it is a recombination. It helps in uh, increasing the diversity of your immunoglobulin molecule. So yeah, that I was explaining right now, that okay, so what RAG does is it will recognize this. What is the work of RAG2? So I think RAG1 and RAG2, they make a complex, and so they make a complex and they work in a like individually. RAG1 is going to recognize monomer, and RAG2 I think is going to cleave it. So RAG1 and RAG2 makes a complex, and then only it can carry on with its function. So okay, somebody has asked what is this IgG binding property of protein A? Explain. I think that protein. So IgG binding property of protein, so whenever like your particular, uh, so let me get a new page. So what happens is your antibody molecule has a heavy chain and a light chain. So the part which is present here, okay, it denotes the FC region. Now this FC region is very, very important in terms of reacting with like proteins of different kind of like macrophage, like this FC region is basically interacting with this protein A, or proteins which are present in like pathogens and uh, pathogens, or any kind of invading microorganisms. So FC region helps in interacting with this particular protein, and then it will help in uh, allowing this protein A to be recognized by macrophages and dendritic cells. The macrophages and dendritic cells will have a better kind of recognition capacity if FC region of your antibody interacts with protein A. So protein A interaction, this is not only just for IgG, it can be for like many other types of antibodies as well. Mainly IgG is more prone to interact with protein A, because during secondary immune response IgG is produced more. So that way IgG can interact with protein A more with its FC region. But according to my understanding, other antibodies like IgE or IgD, this antibodies also interact with protein A, and it helps in coating this protein. Protein A is basically an antigenic protein. So so it can interact with this protein, and then it can present it to macrophages and dendritic cells for phagocytosis. Okay. Do RAG proteins work in humans? Yeah, obviously RAG proteins does work in humans. Otherwise, how are you going to get this many diverse antibody molecules? Because you have so many antigens. You are always in contact with so many antigen, and you always require a repertoire of antibodies in your body. And if RAG proteins do not work, you will not have this many options, this many variety of antibodies that are present in your body. So RAG proteins does work in uh, humans. Madam, what is the difference between the antigen binding capability of TCR and BCR? Uh, so, as I had explained before also, so T cell receptors can recognize antigen only when it is presented by an, one second. So T cell receptors recognizes antigen only when it is presented by MHC molecule. Okay. But B cell receptor can recognize antigen without processing, like just a single antigen it can recognize. Also, T cell receptor requires, co-stimulatory signals like co-receptor binding, CD4 plus or CD8 plus binding for, proper antigen recognition. But B cell receptor does not. It also requires co-stimulatory signals because it has some other receptors, I guess. Uh, B7 or something, I don't recall right now, but it also requires. But T cell receptor, majorly it requires antigen to be presented by an MHC molecule in order to recognize an antigen. B cell receptor does not require antigen to be processed and presented by an MHC molecule. So yeah, that is the major difference between T cell receptor and B cell receptor. Okay, so I'll be disconnecting. Thank you so much.